Hello and welcome back to Biology for Beginners. As always, I'm your host, Morgan. Before we jump into the topic, please remember that Chapter 12 does contain some pretty technical stuff, and the topics here are a little bit heavy and hard to digest, and any average entry-level bio class, most of this chapter would probably be skipped. But the whole march through the book must go on here. So let's talk about DNA profiling. As always, the terms and definitions should be popping up here, so you can pause the video and grab the vocabulary we will be using today. Or if you want the entirety of my slideshow as a PDF, please head on over to my Patreon. The link is down in the description below, and you can grab all of my slideshows and any of my supplemental material for the Bio for Beginners series for absolutely free. However, if you do choose to be a lovely individual and become a patron and make me so grateful, you will get all the Bio for Beginners material at least a day in advance. So without further ado, let's get into this. The first question we need to ask today is, what is forensics? Well, forensics is just the scientific analysis of evidence. And if you're a biologist, the most important application of forensics is DNA profiling. DNA profiling is just the analysis of DNA samples to determine if they came from the same individual or not. And in order to do DNA profiling, you're going to 1, isolate the DNA samples, 2, amplify the markers, and 3, use those amplified DNA markers to compare and decide whether or not those samples are from the same individual. And don't worry, we're going to get into all 3 of these steps. Let's talk about amplifying those DNA sequences. We're going to skip step 1 here because isolating the DNA, for lack of a better description, is just collecting your samples. So let's move on to amplifying DNA sequences. And what you're going to do here is you're going to get a specific segment of that DNA, and you're going to target it and quickly amplify it using the polymerase chain reaction, or PCR for short. And what this does is it just generates billions of copies of that targeted segment, or amplifies. So if we're talking about amplifying the DNA, you're generating copies. And you do this through the 3 repeated steps. And we say they're repeated steps because it's step 1, 2, 3 repeated and multiple cycles. At the end of cycle 1, you yield 2 molecules. At the end of cycle 2, you get 4, so on and so forth. So before we really talk about the steps here, the first thing that you really need to understand is that you need to use these things called primers. Now, primers target that specific segment of DNA that you want to amplify. And the reason they're able to do this is because they're these very short strands of DNA that are complementary to the nucleotide sequence at the beginning and at the end of that segment. So they're going to kind of squash the segment in the middle when they do form those hydrogen bonds with that segment. And the reason you can be sure that they're going to target what you want is because they're chemically synthesized. So they're made in the lab. And that brings us to step 1 of our 3 PCR steps. So at step 1, you have to heat that reaction mixture. And the reaction mixture is what contained your sample. And the reason you need to do this is because, remember, DNA is a double helix. There's 2 strands there, but in order for your primers to connect, you need to get those 2 strands separated, and you can do that by heating the mixture. So in step 2, the mixture is cooled, and the primer molecules are going to be mixed into the solution, and they're going to bind to the ends of the target segment. So instead of the 2 long strands of DNA just sticking back together, you'll get the primers at the beginning and end of the segment you want to amplify. And then in step 3, they use a heat-stable DNA polymerase to then build new strands of DNA by extending the primers. And then you get the macromolecule you're looking for and the first copies of that segment of DNA you want to amplify, and then rinse and repeat and do it over and over again until you have the amount that you need. And again, the DNA polymerase here has to be heat stable because you need to heat and cool the mixture for every single cycle. So once you have all of your target segments of DNA, it's going to need to be sorted. And you're going to sort those DNA molecules by size. And you're going to do this using gel electrophoresis. And gel electrophoresis basically separates macromolecules, and it can do this by size, electrical charge, or other physical properties. But we're going to be talking about size here because we're talking about DNA segments. And so the thing that's kind of the weirdest part in your brain to grasp is going to be the gel. Gel is this kind of thin rectangle of a jelly-like material made from something known as agarose. If you've heard a scientist mention agar, this is what they're talking about. And it's kind of fun to play with in the lab if you get the chance to, but I digress. What it is it's a carbohydrate polymer that's extracted from seaweed. And the specific structure of it just acts as a filter. So this little gel rectangle has wells or holes in the top. And you can place 3 samples in those wells. And then you're going to take that gel and place it in a liquid in a plastic holding tray. And once you do that, you'll put a negative electrode on the end near the wells and a positive electrode at the far end of the gel. And the reason you do this is because nucleotides carry a slightly negative charge and they will move towards the positive electrode. However, because of the structure of the gel, the longer fragments are going to move slower through the gel than the shorter fragments. So you'll get those longer, slower fragments at the top and the slower, shorter fragments closer to the bottom. And when you turn that current off, the DNA fragments can then be stained and visualized, and you can do this by using a radioactive stain or other types of stain, and it will cause them to kind of fluoresce, and you can literally see where the fragments ended up in the gel. Now that we've talked about how to amplify the segments of DNA and sort them by size, let's put it all together and talk about how it's actually used to compare DNA samples. So building those DNA profiles relies on repetitive DNA, and repetitive DNA are just these nucleotide sequences that are going to appear several times in the genome between genes. And one thing about these segments of repetitive DNA is that the shorter segments can repeat multiple times in a row, and these are known as short tandem repeats, or STRs. And different people will have the same sequence in the same place, but with different numbers of repeats. And because of this, we can use the STR analysis method of DNA profiling and actually line those up. And the longer segments will end up higher up in that gel than the shorter segments, making it very easy to tell if a certain sample came from the same individual or not. So in the kind of hypothetical example we have on this slide, you'll see that the top is the crime scene DNA, and there's 2 STR sites. One is a repeat of the segment AGAT, and the second site is a repeat of the segment GATA. And for STR site 1, the suspect DNA matches. However, at Site 2, you'll notice that the GATA in the suspect's DNA is much longer than the segment in the crime scene DNA. So you can conclude that the DNA did not come from the same individual. And that's pretty much how it's used. You just get your primer, grab those sites where the STRs are, amplify it a bunch of times, run it through the gel, and visualize it to see if you have the same profile or not. Now, I know that this is probably less heavy than the last two sections, but this is probably not a section that would be included in entry-level bio class, so we're not going to go into any more detail, but it is a fun little tidbit of information. And I do hope you have a wonderful rest of your day, afternoon, evening, what have you. Please remember to like, comment, and subscribe if you feel so inclined, and I will see you all in the next one. Thank you again for watching this video. Don't forget to like, comment, subscribe, and ring that bell if you would like to see more. And if you'd like to follow me on any of my other social medias, the links are down in the description below. Don't forget to check out thereptilegoth.com for all of my articles and blog posts. If you found any value in this video and you would like to help support the channel, please check out my Patreon page. That link is also in the description down below. And a special thanks goes out to my Diamond Dragon patron, Diane B. What you're doing is really helping me fund a dream here. I will see you guys all in the next one.